Hello, my name is Dan Tempest, and today I'm going to be talking to you about tubular maximum and renal threshold with glucose within the kidneys as it filters the blood. So, first, we need to talk about the importance of glucose. And glucose really comes into the body in the form of our food, and really, most things that we eat is going to have at least some amount of glucose. So, as our food gets digested, that glucose that we eat is going to enter the blood from the digestive tract and it's going to travel to our tissue cells. And that tissue cells, that glucose is going to be used as the main reactant for cellular respiration, where the cells can get ATP, which can do cellular work within the cells. So that glucose is going to travel to those tissue cells via the plasma of the blood. The blood is going to be then filtered at the kidneys, as we see in this image here. So we have the digestive tract where the glucose is going to come from. It's going to travel through that blood plasma, travel to tissue cells like organs, liver, brain, and muscle. And it also is filtered by the kidney. So at normal conditions, the blood glucose levels in the blood plasma is around 70 to 110 milligrams per deciliter. And that's going to be without eating. If you do eat a meal, it should still stay under about 140 milligrams per deciliter. And the entirety of the blood plasma is filtered about 60 times per day. Now, essentially, the higher the blood glucose concentration in the plasma, the more glucose is going to get filtered out. So what I want to talk to you next is kind of where this is going to happen. So again, this happens in the kidney, and it's at a functional unit called a nephron. And we have a nephron zoomed in on, at least part of that nephron is up here. In that nephron, we have a glomerulus, and the glomerulus is where that blood is going to come in. So here we have a glomerulus. There's going to be capillaries in here, and there's going to be some diffusion of glucose, and many other things are going to get filtered out into the renal tubule, which would be this part right in here. Okay. So some of that glucose is going to be filtered into that renal tubule, and the tubule side is of this. So the interior of that tubule is going to have cells called border cells, and those border cells have microvilli, which is like projections into the tubule, and that's going to help to increase surface area, which is going to help increase reabsorption of glucose. This is really important because glucose, as we mentioned, is very important to tissue cells. We need it to be able to do cellular respiration to get energy. And at normal conditions, about 99% of the glucose that is filtered into the kidney is reabsorbed. Now, in order for this to happen, the glucose has to be actively transported into those border cells. So we're seeing some of that right here in the image and right here in the image on the screen. So this needs active transport because we're going from a low concentration to a high concentration. And it actually uses co-transport of sodium ions through the sodium glucose linked transporter, also known as SGLT in the diagram, both the 1 and the 2. Again, that's going to allow that glucose to get into the border cells, and it's going to require some energy because it's going from low to high. Then, once it's in the border cells, it's actually going to diffuse out into the peritubular capillary, and that capillary is going right through here. So here is our diffusion right there, and also in this cell right here. So this is actually going to be something known as facilitated diffusion because it needs this transporter protein in order to be allowed into that capillary. And those facilitator proteins are GLUT1 and GLUT2 as seen again in that image. So that's how under normal conditions, the glucose would get filtered back into the bloodstream. We don't want to lose that glucose. So next I want to go through this tubular maximum graph that also shows renal threshold. So what you're seeing here is a clear linear relationship. So we see that. Let me grab a different color here. Here is our linear relationship that I'm mentioning. This line here that I'm circling. And so as glucose goes up, so does the amount of filtered glucose. So as glucose concentration in the plasma, which is our x-axis here, goes up, so does the movement of glucose, or the filtered glucose. Okay, so we can see again that linear relationship. Initially, starting from bottom left, all or most of the glucose, again, that's about 99%, is reabsorbed. So we can see that again. If we look at this dotted line, that's going to be our reabsorption. And up to about 375 milligrams per minute is our kind of our threshold. You can see up to that point, most, if not all, of that glucose is going to be reabsorbed. Again, that's really important because our cells need that glucose. And no glucose at this time is being excreted. So you can see from this right here up, now we at 300 here, we start to see excreted glucose. But up until that point, if I go draw a straight line down, there is no glucose being excreted. However, there's only so many transport proteins in those nephron border cells within that tubule, and therefore there's going to be a limit to how much glucose can be reabsorbed. Now, under normal conditions in the blood, all of it can be. However, sometimes in some individuals, that threshold can be exceeded. So when we exceed that, you can see our reabsorption stops or levels out. So that limit is about 375 milligrams per minute, and that's going to be known as our tubule maximum. And that is noted on, circling it right now, on the chart. So glucose will be excreted in the urine if we exceed this amount. And this is going to be really just due to the saturation of glucose in those transport proteins. And as we see, that dotted line starts to level off, as I showed. So if glucose levels exceed that tubule maximum, it's going to start to be excreted into the urine. This threshold is called renal threshold, and at 300, and is noted again by the dotted green line. So we see that here. Here's our excreted. And again, that starts to be a thing when we reach 300 milligrams per 100 milliliters of plasma. So this is going to result in something called glycosuria, which is essentially just glucose in the urine. And this is never normal. So when this happens, it can be a trigger. It can be a common baseline test. It's never a sure thing that's for sure somebody has diabetes or something, but it can be something that might be a baseline test to trigger maybe some further testing on diabetes mellitus. So diabetes can cause glycosuria due to the high blood glucose levels and the body's inability of insulin to trigger tissue cells to take it in. Because our tissue cells aren't taking it in, there's going to be higher levels of glucose in the blood. So this is going to result in exceeding that tubular maximum and the renal threshold, and therefore that's the reason why we would find urine in the blood, or glucose in the urine. So we can see again on the left here, we have the normal glucose handling, and there shouldn't be any glucose in the blood, so we see 100% reabsorption. In diabetes, we see the filtered load is going to exceed the reabsorption capacity. Again, we're going to exceed that tubular maximum and the renal threshold, and therefore we have glycosuria, glucose in the urine. Another thing that might cause glycosuria is pregnancy. And in fact, it happens at about 50%. Hormonal changes during pregnancy increases blood flow to the kidneys, causing more glucose to be filtered, and it may reach above those thresholds. This is going to, again, occur about 50% of the pregnancies and can have adverse outcomes on mothers and children if it's not properly checked and monitored. So this is actually somewhat of what the journal article that I looked into studied. They looked at the genetic impacts. So as I mentioned, about 50% of the pregnant mothers are going to experience some form of glycosuria during pregnancy. And this study aimed to conduct a genetic study to find if glycosuria was related to genetics. So they did a genome-wide association study, and it was done on 2 groups, and the results were analyzed. So they used SNPs, or genomic variants, and they found that there was an association with genes on the 16th chromosome, and they found association within that. So this first image, what we have here, is an overview of the whole study. And again, we see genetics were taken from, grab a pen, from 2 groups. Here's the first group, and here was the second group. And down here we can see there was a combined analysis. Okay, so just some other things that we can gather from this. First, this LDSR, that is standing for linkage disequilibrium score regression. So that's going to be done. It was particularly done on the first group. And we also have an ensemble VEP, which was essentially functional informatics, and where we looked at non-code. We actually found that there were mostly non-coding variants outside of that lead SNP. Now, if this column looks at the lead SNP, the one we're really focusing in on, and here's the key result. They found a relationship between the lead SNP and glycosuria determined by this reagent strip that they used. Now, when they combined the results for the 2 groups, they did not find any additional associations outside of the ones in chromosome 16. So both these non-coding variants, which was in this column, and this lead SNP were all found in chromosome 16. The second group was done as like a secondary analysis just to kind of confirm results. So again, they didn't find any new additional associations outside of the chromosome 16 in that secondary analysis. All right. Then this last image, what this shows, first of all, on the left y-axis, we have our p-values. Then we have our recombination rate on the other y-axis. The x-axis has the position of the chromosome. And what I want to point you to is up in here. So this was really the lead SNPs and those associated with that on chromosome 16. You'll notice that they're in a darker red for the most part, and that's going to, if I point you over to this, that's going to be that R squared, and that R squared is really measuring really high rates of linkage. So very connected data up in there. And again, that was our key or lead SNP for this journal article. So essentially what they found, in summary, in their genetic tests of reported glycosuria. So they basically tested a whole bunch of different mothers who suffered from glycosuria during pregnancy, during their third trimester, and they identified associations of genetic variants on chromosome 16. They found that near multiple genes, and they also found that this was implicated in familial renal glycosuria, and that gene was ironically often a target of type 2 diabetic drugs. They also found some linkage to kidney disease, and they recommend further replication of these findings. That would be really important to confirm this data. Okay. So that completes my presentation on glucose reabsorption at the kidney and issues that can go on with that. This first reference is my journal article, and then I have the textbook on here. I hope you enjoyed. Take care.